adaptive immunity with emphasis on a response to SARS-CoV-2 infection. The types of immune responses vary significantly. So we can have a non-specific immune response, which is also called innate immunity or natural immunity. Typically acts as a first line of defense and offers resistance to a variety of microbes or foreign materials. The important part here is that it lacks immunological memory. So there's no specific defense that's happening to a specific pathogen. It's just your body reacting to something that doesn't belong. The main steps in activation of the complement cascade, which are a major component of the innate immunity, are highlighted here. And so over here on the left, you see this pathway of the lectin pathway, as it's called, where these where complement proteins bind to microbes that are coated in mannose, for example, and I'll show you an example of that with respect to SARS-CoV-2. The classical pathway occurs when the microbe is first attacked by antibodies of the specific immune response. Then complement C1 would bind to those antibodies and then create a cascade. And the third version is the alternative pathway in which specific complement proteins called C3b would recognize foreign substances on a surface and bind to those. In either case, it feeds into the production of C3 convertase. C3 convertase then is an enzyme that will cleave C3 into C3b, which will coat additional microbes and continue to activate the alternative pathway. But it also produced this molecule of C3a, which actually acts as a chemoattractant to bring more immune system cells to the fight. This concept of opsonization is important in the sense that when a microbe is coated with these C3b molecules, this microbe is more likely to be phagocytosed or ingested by certain components of the immune system. These little guys act as little handles or receptors that a phagocytic cell can grab onto and more readily engulf this and destroy it. So the C3 convertase and C3b can team up to make an additional enzyme called C5 convertase, which cuts protein C5 into C5a, again a chemoattractant, and C5b, which actually forms this thing called the membrane attack complex, along with a few other complement components. And the MAC is good for punching holes in bacterial membranes and viral envelopes. So SARS-CoV-2 has an envelope. And so the MAC could form in that and cause the contents of the virus to leak out. So here's an overview with specific respect to SARS-CoV-2. The classical pathway is shown here, in which antibodies have been produced against the virus, and that allows the C1 complex to form on the surface, and that in turn stimulates the formation of C3 convertase that you see here. Over here in the lectin pathway, these mannose binding lectins that your body produces will look for mannose residues, and they occur in the spike proteins of SARS-CoV-2, and that activates a membrane-associated protease, serine protease, and that this membrane-active associated serine protease can actually cleave C2 into C2a and C2b, but C4b and C2a together form the C3 convertase enzyme. C3 convertase enzyme in this particular case can cut C3 proteins that are produced and make more C3b, which again acts as that coating on microbial invaders and enhances their ability to be eaten, but it can also work together with a few other factors to make the C5 convertase. And so here's the alternative pathway. And so some amount of C3 is constantly being hydrolyzed, and in this process forming C3 hydrolyzed that you see here. When C3 when C3 hydrolyzed C3 binds to factor B, and then this other thing, factor D, this molecule factor D actually is just a little protease that cleaves the factor B. And so this complex of hydrolyzed C3 and cleaved factor B can work together with C3b here to make the C5 convertase. The C5 convertase in turn can cut C5 into C5b and C5a. And so C5a is actually good for recruitment, just as I had mentioned C3a is for recruiting additional cells to the fight. Now this can be a good or bad thing. If this is overdone, if C3a and C5a are too active, then that can actually cause an anaphylactic type reaction. So C5a will insert itself in the membrane and cause a nucleation, essentially, where the other complement components that I showed you here, C6, C7, C8, and C9 can all assemble and form this pore in the membrane. Now this could be the membrane of an enveloped virus, or it can be the membrane of a virus-infected cell. In either case, cell contents would start to leak out, or virus components would start to leak out and destabilize the structure. This is showing you a little bit of a more detail, but there's one of the aspects of COVID-19 pathology is vasculopathy, where you have these vascular maladies, and they're typically related to complement activation. So in this particular case, here's the SARS-CoV-2 virus binding to ACE2 receptors on an endothelial cell, which would activate complement. Remember that the mannose-binding lectins, for example, could stick to these, or antibodies that are produced might activate the classical or alternative pathways. And that's what you see here. The lectin and classical pathways being activated, that leads to the formation of C3b. And again, that tick over event can lead to more alternative pathway activation, the formation of C5 convertases, which then cleave C5 into the chemoattractant, and then the first part of the membrane attack complex, C5b. And so C5b deposits on the, and imagine in this particular case, an endothelial cell membrane. So these endothelial cells would be cells that line a blood vessel, for example. So imagine then C5b inserts, and then the other complement components, C6, C7, C8, and C9 form the pore. In turn, that pore formation will cause damage to the membrane. And so that stressed cell will start to produce chemoattractants like MCP1 or monocyte chemoattractant protein. And then they'll also produce interleukin 8, which is a cytokine, which can trigger more immune system cells to come to the fight. In this particular case, a neutrophil called a PMN. And so these PMNs can then attack the cell as well and pump them full of reactive oxygen species that can destroy these. And so again, imagine if this is a blood vessel and this is an endothelial cell lining the blood vessel. These neutrophils can start to attack this blood vessel then. So the additional things that can happen in this particular case is these, you see these E-selectin, you see these P-selectins, these ICAMs and so forth. These kinds of molecules that form on the surface actually allow other cell types to stick to those cell surfaces. And so that causes a recruitment of immune system cells, and it also triggers some of these cells to additional cells to come punching their way through into the tissue to fight whatever's out here. And so when these cells start punching their way through and binding to these surfaces, they can start damaging endothelial cell membranes. So as I said, the MAC formation can happen, and that can cause cell leakage. And the cells themselves start to produce these VWF, for example, von Willebrand factor or P-selectins, which cause platelets to aggregate on the surface. And that's part of a clotting cascade. You see a membrane attack complex forming inside a platelet, which then causes the platelet to start releasing these PMPs, adhesion molecules. These coagulation molecules, and then this process here, the VWF, P-selectin, and platelet aggregation, can lead to the secretion of thrombomodulin that you see here. This particular protein from the surface starts sloughing off and interacting with PMPs that are leaked out of platelets, and that causes a coagulation cascade. And so again, coagulation maladies are part of COVID-19, causing blood clots, for example. And so this whole process of MAC formation and platelet aggregation and coagulation can lead to the inflammatory vasculitis type terrible outcomes that we can see in certain cases of COVID-19. One of the important components of the innate immune response is the activity of interferon. I should back up, actually. I forgot to mention this, but complement proteins are naturally produced by your liver, and so they do a lot of good things. So this happens to be some bad things that can happen, but as I said, complement C3b can coat organisms and facilitate their uptake and destruction by phagocytic cells, and obviously recruiting components like C5a and C3a are important for bringing other immune system cells to the fight. And so they do have essential functions in clearing infections. And so a vigorous innate immune response can be very important. And it's probably why children, for example, are not as susceptible to COVID-19, because they have a vigorous innate immune response that's well balanced. And so they rarely show symptoms. And so another component of the innate immune response is interferon. And so in this particular case, interferon works as a signaling molecule, an alarm molecule. And so here you see an infected cell. This infected cell, here's a virus, and the virus has inserted its genetic information into the cell. And the genetic information encodes new viruses, which bust out into the world. But before this can happen, the virus RNA sets off a cascade of events. So the RNA here interacts with an adapter protein and some of these initiation factors that ultimately activate transcription factors like NF-kappa B. And so NF-kappa B will activate interferon production and a protein kinase called protein kinase R, which phosphorylates elongation factors in the cell as well. So phosphorylated elongation factor in this particular case will actually slow down ribosomes so they won't translate viral proteins as fast. But interferon's job is to signal neighboring cells to warn them. And so this warned cell then will produce high levels of RNase, for example, an RNA digesting enzyme. So if this virus comes and tries to drop its RNA into the cytoplasm to synthesize viral proteins, the RNase will attack it and chop it to pieces. The cells also produce RIG1, which is another RNA sensing, virus RNA sensing protein that also leads to interferon production. And so again, it's really important to produce this warning signal in order to get this cell primed and ready to fight this virus. Because if the virus comes before this signal, then the virus will win. It's faster. But if the nearby cell has a head start on producing these activities, then it can meet the virus RNA with a little RNA degrading enzyme on the front end. So natural killer cells are parts of the innate immune response as well. These guys they're a small population of large non-phagocytic granular lymphocytes, meaning that they contain granules that are full of toxins that will kill virus-infected cells. And so you can imagine these guys being very important in fighting infection. They produce a couple molecules that punch holes in the cells and actually kill them. But what's interesting is natural killer cell activity decreases with aging and obesity and some of these things that we find as comorbidities in COVID-19. So it's not surprising then that individuals that are older or obese or have other maladies are more susceptible to COVID-19 because of the natural killer cell dysfunction that's associated with their condition. And we know that COVID patients that are really sick have low levels of circulating NK cells compared to people that only have mild disease. So that suggests that the virus probably needs to dampen the NK activity in order to really succeed at affecting the host. And it also suggests strongly that we probably should have a way to detect NK cell function, or at least to use assays that detect NK cell function. That probably could be useful in determining what kind of outcome an individual might have in an infection. So again, there's two ways that NK cells recognize target cells. They can bind to antibodies that are coded on these cells, or they can recognize the loss of the MHC antigen on the surface of the cells, which is typical in virus-infected cells or cancerous cells. So that looks something like this. So here's the natural killer cell, and it's basically doing a handshake, just kind of feeling around and doing a handshake on a normal cell. And so this guy has a normal MHC class 1 molecule on its surface and some other ubiquitous receptor. So this handshake would be successful, and the natural killer cell would leave it alone. But imagine this guy now is a virus-infected cell, and it's abnormal because it's not displaying the MHC class 1 molecule. So that would immediately trigger this guy to attack and basically produce perforin. Perforins just punch holes in mitochondrial membranes and the cell membrane itself, and then produce these granzyme type enzymes, which will damage DNA and so forth. And so that will, basically in general, that will cause the cell to undergo a process called apoptosis or programmed cell death. So that's pictured here. In this particular case, the NK cell has antibody receptors on its surface, and so antibodies that are produced to a virus antigen on a infected host cell would bind to these guys. And so here's the virus-infected cell. And one thing that virus-infected cells do is they display antigens. They display pieces of the virus on their surface, and that would trigger an antibody response. And so that's what's happening here. And since this NK cell has antibody receptors on its surface, it can bind to that more efficiently and then pump this guy full of toxins to kill it. Obviously, you don't want NK cells just running around killing things, normal, healthy cells in your body. So there has to be a way for these guys to triage and figure out who's healthy and who's not. And this is one way that this happens, having antibodies on the surface that are bound to virus antigens that are being displayed. And so again, we look at this sort of low-risk individuals versus high-risk individuals and outcomes for these guys. And so there's a healthy population of NK cells. Here's a virus-infected cell. The NK cells will recognize certain antigens that are displayed, virus antigens that are displayed on the surface, and then bind to those or bind indirectly to those by antibodies that are on the surface, rather, and then pump the cell full of granzymes and perforins to clear the infection. In so doing, they also trigger the formation of interferon gamma, for example, which is a molecule that will act as a chemoattractant and stimulant to other components of the immune system to come fight the infection. But in general, this is all done in a controlled way. And so we don't have excess damage, we just clear the infection. With high-risk individuals, there's a dysfunctional NK cell response, and as a result, the NK cells grow like crazy. They're less able to recognize the distinction between an infected cell and an uninfected cell, and so they start to produce excess cytokines and chemokines. They produce a ton of interferon gamma. That triggers a lot more cells to come to try to fight, and an overabundance of cytokines, chemokines, and so forth. And there's the sort of the basis of that cytokine storm. And so not only are the viruses destroying cells, but your own immune response is destroying the cells as well. And as a result, you end up with the acute respiratory distress syndrome, or ARDS, that's typical of bad outcomes in COVID. This is an important comment from a particular paper. NK cell levels in peripheral blood inversely correlated with disease severity. And so again, it's not just the numbers of NK cells, it's also how well regulated their activity is. So types of immune responses: in addition to the non-specific or innate defenses, there's also specific immunity. It's also called acquired immunity because you're acquiring it specifically to fight particular pathogens, or adaptive immunity is also used, and again, specific immunity. It gives you resistance to a particular foreign agent, and the key component here is that it has immunological memory, meaning that the effectiveness of the attack increases upon repeated exposure to a particular agent. So if you've had measles once, you may get sick, your body fights the measles, you recover, and then the next time you get measles, the reaction is so rapid and so efficient that you never even develop any symptoms. The two types of responses, not surprisingly, usually work very well together to control infection.